T-cell story, and indeed it's better to say I'm going to apply cellular immunity. Can we say that T-cells are only involved in cellular immunity, or in other words, can I say that T-cells do not have any role in humoral immunity?
T-cell
Have role. Class switching, affinity maturation. If a patient cannot produce MHC protein, what will happen to that patient? A patient who doesn't express any MHC. Severe immune deficiency, yes. What happens for its cellular and humoral immunity?
We won't have the T-cell and B-cell, or we won't have activation of T-cell.
He won't have activation of T-cells?
We will have B-cell, B-cell, that's like low affinity, we don't have like normal B-cells, and T-cell we don't have that T-cell because we need that.
Yes. So when there is no MHC, there is no activation of T-cell. When there is no activation of T-cell, we won't have T-cell help for B-cells. So both arms of the immune system will be very weak. So the patient will have severe immune deficiency. So that's why I'm saying that T-cells are not only involved in cellular immunity. We have their role in humoral immunity as well. Do we need APCs for humoral immune response or not? Do we need APCs for humoral immune response or not? Yes or no?
Yes.
She says yes. What about the rest of you? Do we need APCs for humoral immune response or not?
B-cell is ABC itself.
Yes. What else? B-cell is APC itself. What else? APCs have another role.
We need ABC to activate T cells.
We need APC for activation of T cells, and we need T cell help for B cell again. So I told you there is a network of cells always. There is indeed a network of cells for B cells, T cells, APCs, cytokines. Many proteins and cells are involved in each immune response. So again, T cell story. T cell development occurs in the thymus. The process of T cell development is similar, is the same process for B cell development, and the process of production of different BCRs, diversification of these TCR is same as BCR development, so I'm not talking about them again. And it occurs in the thymus, primary lymphoid organ, T cell activation in the lymphoid tissue, secondary lymphoid organs, and function of T cell is different. T helpers mainly in the secondary lymphoid tissue, but cytotoxic T cells go to the site of infection and directly fight with the tumor or infectious cells. So T cell development, again, a T cell has several TCRs, and all of them are against just one epitope. They have same specificity, and this is the structure of a TCR. TCR is made of two chains. In the antibody, we had 4 chains, 2 identical heavy chains and 2 identical light chains. But here we have just 2 chains. Okay? Mostly the TCRs are alpha beta TCR. It means that we have a chain of alpha, another chain beta. It's the name of the chains. And the variable regions again will make a site for antigen binding, which is called antigen binding site. Each TCR has only one antigen binding site, not two antigen binding sites. It's different from antibody. Antibody has two arms, but TCR has just one arm and one antigen binding site. So here is the antigen binding site of these TCRs. So the majority of T cells in the body are called alpha beta T cells. Alpha beta T cells mean the T cells that their TCR is made of alpha and beta chains. But we have two types of alpha beta T cells. One of them are CD4 positive, they are CD4 proteins. They are CD4 proteins. And another one is CD8 positives. They are CD, and you know that CD4 positives bind to MHC class 2 on the APCs, but CD8 positives bind to MHC class 1. This is mainly against intra-only cytotoxic T cell. CD8 positive T cell is indeed cytotoxic T cell. Another name for it is CTL. So CTL is only against intracellular antigens, only and only. It can do nothing against extracellular antigens. Okay? Intracellular viral antigens, tumor antigens, any intracellular, and mainly cytosolic antigens. But CD4, we have different types of CD4 or T helper cells. T helper 1, we have talked about them. T helper 1 provides help for CD8 positive cells. T helper 2 will produce other cytokines. T helper 17, I will talk about them, and mainly the other one is not and regulatory T cells. Regulatory T cell is not helper T cell. It's a CD4 positive T cell. It's not a helper T cell. It's just CD4 positive T cell. A minority of T cells in the body, they have gamma delta receptors. The receptor is different. Instead of alpha and beta, they have gamma and delta. So they are called gamma delta T cells. Okay? Gamma delta T cells are the minority of the cells of T cells of the body. But alpha beta T cells are the majority of the T cells of the body. And gamma delta T cells do not need MHC for activation. In opposite to alpha beta TCR or alpha beta T cells that they need MHC, gamma delta T cells do not need MHC, and they have different receptors. Some properties of gamma delta T cells is similar to innate immune cells. They have PRRs, you can see them here. They have TCRs, and they do many things. And they are one of the important cells of mucosal barrier or epithelial barriers. I'll talk about it in the barrier lecture. So gamma delta T cell is abundant in the barriers, and it provides a rapid immune response like innate immune cells. It's have some properties of adaptive immunity, TCR, production of cytokine, and some properties of innate immunity. That's enough for gamma delta T cell. So the gene of alpha and the delta, gamma, and alpha beta is different. So the diversity of T cell, like B cell, is we have again variable regions, antigen binding sites, and again the story of genes, chromosome, genes, proteins, the limitation of this story for production of millions of receptors. So we need diversification process. I'll escape from them. Having fragmented genes, then all the process is as same as B cell, but the genes are different. For B cell, we have immunoglobulin genes and their fragments involved. Here we have alpha and beta chains and their genes are involved. Again, as similar to immunoglobulin genes, variable regions, that they are fragmented genes. The variable region of TCR genes are fragmented as well. Okay, so we have fragments and multiple versions of each fragment, and then we will have different combination of each fragment. And again, we have V, J, and D segments or fragments, and then binding of different fragments to each other and making different combinations. And again, so RAC enzymes are involved here again, the story is saying. And again, we have junctional diversity to add some nucleotide in the binding site of the fragments, and increasing the diversity of TCRs. The story is saying, I won't explain them again. If you have question, I can explain. If you do not have question, there is no need to explain them again. The story is saying. So development of T cell that occurs in the thymus, but the precursor of T cell is produced in the bone marrow, and the precursor or progenitor of T cell moves from bone marrow to the thymus, and the thymus it will produce in sequential stages to develop mature T cells. That mature T cells is called naive T cells. You know this, and you do not need to memorize the content of the table here. Let me skip. So in the thymus, we have again cortex, medulla, and I told you that we have a rapid proliferation of the T cells in the cortex of the thymus, and here we have the cells are not as dense as the cortex because most of them are eliminated. I'll talk about them in the tolerance lecture. Let me skip. So that's enough for T cell development. The story is the same as B cell. But for T cell activation, the story is different. We have two types of T cell, alpha beta and gamma delta T cell. Alpha beta are called conventional T cells, and they are the majority of the T cells, but gamma delta T cells are the minority of T cells of the body, and their activation is different. Alpha beta T cells are mainly in the lymph nodes and spleen, but gamma delta is mainly in the mucosal surfaces. I'll talk about them. So, after development of T cell and going to the lymph node or secondary lymphoid organs, we need antigen presentation by mainly dendritic cell. Okay, and dendritic cell has a very potent APC. It will present antigen to the CD4. This one is called MHC. As the T cell is CD4, so it's class 2 TCR MHC. And this one is the yellow one, peptide. Yes, antigen. And this one, TCR. So this provides the first signal. Is that enough for T cell activation? No. We need co-stimulation as well, but I have another slide for it. So after activation of a naive T cell, we will have again clonal expansion. We are talking about activation of CD4, clonal expansion, and differentiation to different T helpers. We have other T helpers, including T helper 22, T helper 9, but they are the most important of T helpers. So T helper 1, 2, 17, and regulatory T cells, all of them are CD4 positive T cell. And a small amount of the activated T cells will differentiate to memory T cell. So this one is called naive T cell, but those are effector T cells. Before activation, this cell is called T helper 0 as well. T helper 0 is a naive CD4 positive T cell that it hasn't been differentiated to each of these different T helpers. For CD8 activation, we need MHC class 1, and of course we need co-stimulation. I have another slide for this, I've told it several times, co-stimulation. And of course, a dendritic cell are present in antigen to CD8 and CD4 simultaneously. With its MHC class 2, it activates CD4, and with its MHC class 1, it activates CD8. And then we will have again clonal expansion of CD8 and differentiation to CTL, cytotoxic T lymphocyte. And then this CTL can recognize the MHC class 1 and antigen in the cell again. This one. So this MHC class 1 and peptide is presenting by DC. Here the antigen is presented by infected cell. But here we need co-stimulation because the cell is naive, but here we do not need co-stimulation because the cell is an effector cell. I've told you that for naive T cell we need co-stimulation. Or CTL can recognize the tumor. This is a virus infected cell, this one is tumor cells. And after activation, I'll talk about it, and some of them again will differentiate to memory cells. All of this process occur in the lymph node or secondary lymphoid organs. Again, we have cortex, paracortex, afferent lymphatic vessels, efferent lymphatic vessels, and entrance of B cells and T cells, B cell zone, T cell zone. And in the spleen we have again B cell zone and T cell zone. You do not need to memorize this image and this one as well. In the spleen again we have T cell zone and B cell zone. And after activation, here we have activation of CD4 T cells. This is an APC and they are antigens, that they are phagocytosed by antigen and presenting cells, APC, and some part of the antigen is presented by MHC class 2. And then we have CD4 positive T cells and CD8 T cells. Only CD4 bind to it, and then there is co-stimulation by B7 and CD28. B7 is on the APC, CD28 on the T cell, and after activation, T cell will produce IL-2, interleukin 2. Interleukin 2 is a very important cytokine in the clonal expansion of T cells. Clonal expansion of T cells need production of IL-2. So then we will have clonal expansion or proliferation of the clone, the cells of the clone, and even this ABC will produce some cytokines, and based on the type of cytokines, this CD4 positive T cell will produce two signals for T cell. First one is CD40 and CD40 ligand, and the second one, different cytokines. Based on the type of those cytokines, we will have different CD4s, and these CD4s will produce different cytokines. I think everything is clear, yeah? I've talked about them several times. So, here is, for example, when an APC produces IL-2L, we will have T helper 1. And T helper 1 will produce interferon gamma. I've talked about it. Interleukin 12 is very important in the differentiation of T helper 1 and cellular immune response induction, and interferon gamma is macrophage activating cytokine. And of course, interferon gamma can induce class switching in B cell for IgG. And if interleukin 4 is in the environment of the T cell, it will differentiate to TLPER2, and TLPER2 will produce interleukin 4, and this interleukin 4 will act on B cell and induces class switching to IgE. So based on the type of antigen, we have different helper T cells, different cytokines, and different classes of the antibody. You can see the network again here. And if the cytokine is TGF-beta, we will have regulatory T cells, and as a result of binding of TGF-beta to the B cell, we will have IgA production. Okay? Another cell, TLPER17, when the APC produces some cytokines together, we will have TLPER17. TLPER17 will produce two important cytokines, interleukin 17 and interleukin 22. Interleukin 17 is very important in the activation and recruitment of neutrophils, and neutrophils are important in extracellular fungi and bacteria infection. And interleukin 22 is very important in barriers, mucosal immune surfaces. Okay? So, activation of CD8 positive T cells and MHC class 2 for CD4 positive, production of interleukin 12, and so we have an IPC which is presenting by MHC class 2 for CD4 positive T cell and co-stimulation as well. Co-stimulation as well. On the other hand, MHC class 1 and co-stimulation for CD8 T cell, and then CD4 will produce interferon gamma to increase the activation of TLPER1. I've told you that is very important in cellular immune response. So TLPER1 will produce interferon gamma to increase the activity of CTL. And then we will have activated CTL, and this CTL without any need for co-stimulation can recognize the target cell. It does both tumor cells or virus infected cell, and it will release the content of its granule. What is inside the granules of the CTL? Perforin and granzyme. The content of the granules of CTL and NK cell is same. Both of them have granules, and inside the granules there are perforin and granzyme. Perforin makes hole on the membrane of the cell, and granzyme induces apoptosis, the death of the target cell. So we have the death of the target cell, and that was this one was virus infected cell. This one is tumor or cancerous cells and cancer antigen, and killing of tumor cell by CD8 T cell. CD8 T cells can use another mechanism that I'll talk about it in the next lectures as well to kill the target cell. It means binding of Fas ligand to the Fas on the tumor cell. Fas is a death receptor. In opposite to the co-stimulator and co-receptors, Fas is a death receptor. After binding of Fas ligand to the Fas, this receptor induces activate apoptosis in the target cell. So CD8 positive T cells use two mechanisms to kill the target cell. The first one is using the content of its granules, perforin and granzyme. The other one is using Fas ligand and Fas. Clear? That's it. There is a sample question here. Have a look here. You've had the question, I think.
C.
This one is not function of CD4 T cell. Yes, tumor lysis. CD4 T cells can help B cell for class switching, can help for CD8 maturation. It's better to say it's instead of writing maturation, it's better to say activation. Have a look here. CD4 T cells do not have any role in maturation of CD8. It has role in activation of CD8, not maturation. Maturation occurs in the thymus, and CD4 positive T cells have no role in the maturation of CD8 T cells. It has a role in the activation, not maturation, and lysis and enhancement of killing properties or capacity of the macrophage. That's it.